Okay, good afternoon everybody. Welcome to today's webinar from CHCS, the Chemical Hazard Communication Society, on in vitro skin corrosion testing. So I'm just going to have a couple of, okay, sorry, I gather I'm perhaps showing you the wrong screen here, just give me a second. Right, I hope I'm now showing you the right screen. Okay, so welcome again then to today's webinar from CHCS on in vitro skin corrosion testing. So my name is Jill Paluka. I'm the chair of the CHCS and I'm pleased to be chairing your webinar today. Before we get started, I would just like to go through a couple of things just to give you an introduction to the GoToWebinar software that we're using today if you haven't used it before, and just to talk to you about, just to tell you how we're going to manage questions, and then some follow-up and feedback after the webinar. So first of all then, to participate today, if you should be able to see in the top right hand corner of your screen, there will be a little box with a red or green arrow. If you can't see the box, if you just click on the little arrow, it'll open up. Once you have got the box open, then you'll be able to see there are various options there which allow you to manage your audio, so you can view that, select that, test your audio. You can also open and close the panel using the little arrow at the top corner there. So that shows how that happens. If you have any problems today, you can raise your hand, and our website, the webinar administrator, Dave King, will contact you via text message using the chat facility, which you can see at the bottom. You can also submit your questions to us, so we will be taking questions at the end of the webinar. If you think of a question as you're going through, then please do feel free to type that in as we go along, and of course you can also submit further questions at the end of the presentation, and we will then ask Carol to answer those questions for you. Okay. After the webinar, we will send you an email with some links. We will, as always, from CHCS, send you a feedback survey. We really do appreciate your feedback on the webinar today, and also anything else you want to tell us about CHCS, so that we can continue to develop and improve these types of events. And particularly if you've got any ideas for any additional events that we aren't running at the moment, then please do give us your ideas, and then we can try and make sure that we are providing the information that you as our members would like to see. We'll also send you a link to a PDF copy of the slides and to a recording of this webinar in case you want to play it back and listen again. So your webinar presenter today is Dr. Carol Treasure, who is the CEO and founder of Accelerate. Accelerate conduct regulatory in vitro safety tests for a variety of chemical industry sectors, and the company's work has been regulated at a regulatory level, recognised at a regulatory level by the OECD and by top funding bodies such as Innovate UK and Horizon 2020. Carol holds a first degree in physiology and pharmacology from the University of Sheffield and a PhD from the Frame Alternatives Laboratory at the University of Nottingham, based on skin cell biology and reconstructed skin models for in vitro safety testing. She has devoted her career to using innovative science to replace animal testing over the past 25 years, working closely with leading cosmetic and chemical companies and their supply chains. So Carol is very passionate about the use of in vitro skin testing, which I'm sure was going to come across in the presentation. So Carol, then, without further ado, I'd like to hand over to you now.
Okay, thank you very much, Jill, for the invitation to speak today and for that introduction. It's great to have you all with us to discuss in vitro skin corrosion testing. I hope you're going to find this useful, and as Jill said, please don't hesitate to type in questions as we go along. I'll try to answer as many as I can at the end, and perhaps we can also send round some written Q&A if we don't manage to get to them all today. So I'd like to talk about in vitro skin corrosion testing. Just trying to move to the next slide. Okay. I know that we have quite a wide variety of experience levels participating today. So if you know some of the content of the early slides, then please bear with me. But I thought it might be useful just to start with some basic definitions, just to set the context for the rest of the presentation. Sorry, I'm finding that each slide is not loading. Of course, it was fine in the practice. You just bear with me. I'm going to try coming out of the presentation mode and just going into alternative view. So I'm sorry about that, but I hope you can still manage to see the content. So if we just start with some definitions of skin irritation and corrosion and think about what the differences are, the main difference is that dermal irritation is the production of reversible damage to the skin, and that's following application of a test chemical for up to 4 hours. And by contrast, dermal corrosion is the production of irreversible damage. So we'd be looking at things like necrosis of the epidermis moving down into the dermis of the skin, again following application of the chemical for the same period of time of up to 4 hours. And this is defined from traditional animal studies, and corrosive reactions are considered to be typified by things like ulcers, bleeding, scabs, and after the 14-day observation period, you would get discoloration due to blanching of the skin and complete areas of alopecia and scars. So there's a very strong difference between irritation and corrosion, with corrosion being irreversible. And the other definitions I just thought I would show for clarity before we move on to talk about testing are the GHS categories for skin corrosion and irritation. And these have been defined according to the animal studies, which would have been using Test Guideline 404, which is the Dray's test. And that's why there's references here to the animal responses. So category 1 is corrosive, and that describes the destruction of the skin tissue in at least one animal after exposure of less than 4 hours. And in cases where it's not possible to subcategorize into 1A, B, or C, there's a standard definition that you would just use category 1. But you can see that following on from that, there are subcategories A, B, and C, which are progressively weaker responses. And so 1A means that there would be some kind of response in less than 3 minutes. 1B, there would be a response between 3 minutes and 1 hour. And 1C, the response would take place after 1 hour, but before 4 hours. And as we move on to talk about the in vitro tests, you'll see that they've been developed with these time points in mind so that they can model the subcategories that were originally described by the animal test. And then, of course, moving on, we have the GHS category 2, which is a skin irritant as opposed to a corrosive. And then we have no category for those not causing any effects. I think that the easiest way to think about the process of assessing skin corrosion and irritation is to follow the OECD guidance document 203, which was published back in 2014. and that describes an integrated approach on testing and assessment known as an IATA, which I'll talk about quite a lot today. This particular one is for skin irritation and corrosion, but there are other IATAs that have been published as well over the last few years looking at different endpoints, and they take the user through an overall approach so that existing data can be assessed, and then you can decide whether any additional testing is required. So the IATA has three parts. Part one is a review of existing data. Part two is a weight of evidence analysis using that data. And then part three is the performance of any additional testing to generate more data if that's needed. I have included a link to the IATA online, so if you're going through this afterwards, it's well worth taking a look. So I've just zoomed in here on parts 1 and 2, and it just describes in a little bit more detail the steps that are required to analyse existing data. So the type of data we'd be looking at is any existing skin irritation and corrosion data, as well as any other data from other dermal toxicity methods, and along with physical chemical properties of the chemical and any other data that has arisen from any non-testing methods. And from that, you would go on to create a weight of evidence on a response that you would expect to see. And at this point, it's quite common to build in what are described as analog approaches, such as read across from similar chemicals, to create an overall weight of evidence to decide how likely it is that you would see a corrosive or irritant response, and to understand at that point whether we have a conclusive assessment of the corrosive potential, or whether it's going to be necessary to go on to any further testing. Now, part 3 is actually the main focus of the presentation for today, and I'm just going to go up to presentation mode for this particular slide because I want you to be able to see it as large as possible. Once we've identified the most likely outcome of the test based on our weight of evidence, that then dictates the testing approach to take. And if we're expecting the chemical to be an irritant or corrosive, we would follow what's known as the top-down approach, whereby we start with a skin corrosion test, and that would be the in vitro test that I'm going to go on to describe shortly. If we're expecting the chemical not to be an irritant or corrosive, we would go for the bottom-up approach, which actually starts off with a skin irritation test and only goes on to a skin corrosion test if required. So this is partly designed to limit the number of additional testing that is needed by making an educated guess at the outset whether you need to start from the top end of corrosion or the bottom end with irritation, and you may not necessarily have to go on to do the second test if you get a conclusive result in the first one. And I just thought it might be useful to take a look at this, this data. This is taken from an ECHA summary report back in 2017, and it was a summary of the options that have been used by REACH registrants up until that date. So I've highlighted in the top row the skin irritation and corrosion data. And if you just take a look at the key at the bottom of the slide, we can see that the vast majority of cases were able to be resolved and assessed using data from old experimental studies, read across, or weight of evidence. But there was still around 20% of those cases that required additional testing, and that would have been in vitro testing wherever possible, and possibly with just a small number having a requirement for in vivo testing as a follow-up. And we'll go on to discuss that. So I'd like to get into the sort of main part of this presentation today, which is to talk to you about the skin corrosion tests, which are described and approved in the OECD guidance document. There are 3 tests which have been defined in their own OECD Test Guidelines, which are referenced in the IATA. And the first one is Test Guideline 431, which is a human reconstructed epidermis method. The second is Test Guideline 435, which is the in vitro membrane barrier. And the third is Test Guideline 430, which is the transcutaneous electrical resistance method. So what I'll do is I'll just work through those 3 tests and highlight the key points about those and things that we need to consider if we're thinking about performing these particular tests. One of the best known is OECD Test Guideline 431, which is the reconstructed human epidermis method. And this was actually one of the first in vitro tests to gain full regulatory OECD approval back in 2004. So time's flown, that's 16 years ago now. And it's updated on a regular basis, so it is important to keep checking that you're working from the most recent version of the OECD guideline. Now, the scientific rationale behind this test is that corrosive chemicals cross the skin barrier either by diffusion or by erosion, and that when they cross that barrier and the skin cells underneath are exposed to that chemical, that the chemical is toxic to those underlying cell layers. This provides the closest possible simulation of real-life human exposure. This is the closest that we can get without doing human trials, which clearly wouldn't be ethical for an endpoint like this. And what we're doing is using essentially a little piece of tissue engineered skin in the laboratory, and it has a very closely aligned morphology and function, just like human skin, and it has a barrier function or a stratum corneum, which is critically important when looking at chemicals which may get absorbed across the skin and have a corrosive or irritant effect. This model is suitable for substances and mixtures, and it's used regularly for solids, liquids and emulsions. And the method involves a topical application of the chemical or product onto the surface of the skin for 3 minutes and 1 hour. So just thinking back to the definition slide, these were the time points that were originally used in the Drey's rabbit test, and that's why they were chosen in parallel to develop this in vitro method. You'll also see that there's a 4-hour additional time point for the episkin method, which is used to subcategorize chemicals, and I'll go on to talk about that in detail in a moment. Now, when the chemical is applied to the skin, the cell viability is then measured by metabolism by those cells of a tetrazolium salt known as MTT, which is really widely used as a viability indicator. And MTT is converted to a blue formazan product, which can easily be quantified by spectrophotometry. So essentially, if the cells are damaged, we're getting a smaller amount of the blue formazan product, and that's reflected in the reading of the spectrophotometer compared with the untreated control. So corrosive chemicals are being identified by a decrease in cell viability below well-defined threshold levels that were identified during the validation study. Now, there are 4 models that have been validated and permitted for use with this method, and they're provided by a variety of commercial suppliers. This can seem a little confusing when looking at the test guideline, but it is really important to think about what your desired outcome is, what type of data you need, and therefore which is the best skin model to use. So I'll go on to those in a bit more detail on this slide. I think there are a number of factors to consider. These can include the predictive capacity or subclassification that you need. Do you just need a straightforward category 1 versus non-corrosive? Or do you actually need to know what the subclassification is for your chemical? And you may have an indication of your expected result through your weight of evidence approach. I think also a lot of companies have gathered historical data over the years using a particular skin model, and then it can be considered a little bit inconsistent maybe to change, or there might be a reluctance to change if you're very familiar with one model and you know that it works well. I would say that even though all 4 of these companies do ship around the world, they all ship internationally, but there are some limitations on accessibility depending on where you are, and there are also price implications as well. So I think this is a really important factor to discuss with your test lab before embarking on the testing, and it's about getting a balance so that you definitely get the data that you need, especially if you need subclassification, but without going overboard in terms of price, etc., if it's more than you actually need in that case. So you can see that the 4 skin models, they've all got good morphology that is very close to human skin. They've all got slightly different characteristics, and they've been stained with slightly different dyes, some of them at the bottom here. So it's not an exact comparison, but these are the photos that I was able to get for today. And you can see that the thickness of the skin barrier also varies between some of the models. And so if we think about the performance of this in vitro test, these figures were generated from the EpiSkin skin model, which was the first one to be validated, and compared with the traditional Dray's test. And we'll look at these together when we look at the other methods at the end of the presentation. But just to say for now that it did have a strong performance compared with the Dray's test, a sensitivity of greater than 95%, a specificity of greater than 90, and an overall accuracy of greater than or equal to 82.5%. So if we're thinking about which skin model to use and thinking about the predictive capacity that we need, I've tried to just summarize quite a complex situation in a few bullet points here. And I think the important points to remember are that all 4 of the RHEs are successful in discriminating skin corrosives category 1 from non-corrosives, but the EpiSkin model is able to discriminate between subcategories 1A and 1B or C. Now note that doesn't mean that it can tell 1B from 1C. It can only subcategorize whether it's 1A versus 1B or C, and it has formal approval in the test guideline and the IATA for that process. Now, EpiSkin can also discriminate between 1B and C, but doesn't have formal approval. So this is where it might seem a little confusing. So 1A versus 1B stroke C has formal approval, but if you want to discriminate between 1B and 1C, it doesn't have formal approval, but a lot of validation has been done to suggest that it does have the potential to do that. So there are published studies which are quoted in the IATA, which are very helpful to look at, and it's recommended that those are used for a weight of evidence in order to avoid the need for additional animal testing. So I think that that's a useful thing to bear in mind depending what you need for your chemical. And then the epiderm, skin ethic and the CHCS models are all currently accepted to identify 1BC from not further resolved corrosives. So you would really only use those if you didn't have a strong need for subcategorization. Now, if we just consider the strengths and weaknesses of this model, the strengths are certainly that this is a model that's been around for a long time, for 16 years. There's now a lot of historical data. It has formal validation and OECD approval. It has widespread recognition by regulatory authorities around the world. And it's very highly relevant physiologically using reconstructed human tissue. And also knowing that the partial subcategorization is possible, that's a distinct advantage. It does have some weaknesses, and it's important to keep in mind that some coloured chemicals may interfere with either the metabolism or the detection of MTT. And so preliminary steps are built in prior to actually getting on with the test itself, just to identify any chemicals that might cause interference. But the good news is there are additional controls that are built into the test to make sure that we're taking account of that. Now, this test is not validated for gases or aerosols. You'll see as we go through that actually none of the tests are either in vitro or in vivo. I think there's been some research in that area, but it's not formally validated. Also to keep in mind that the subcategorization aspect does have a tendency to overclassify into category 1A. and the IATA states that that was varying between 21 to 46%, depending on which skin model was used. So check out the IATA for more information on that. On the one hand, it's a good thing because it's erring on the side of caution. It's actually defaulting to 1A, which is the more aggressive corrosive category, so it's erring on the side of safety. But that can still be an issue if it's over classifying in terms of labelling and packaging, etc. So the limitations of this method, none of these skin models may formally be used to distinguish 1B from 1C. I think it's interesting to note that the reason cited for that is that there's currently only a very limited set of well-known in vivo corrosive chemicals available for reference for validation. But that wasn't raised for the other guidelines, so I think that there's still some debate to be had there. And in common with the other methods, this method doesn't discriminate skin irritants from chemicals not requiring classification. So it won't tell you whether it's Cat 2 versus no category. But for that purpose, an additional guideline is available, which is Test Guideline 439. I'm just going to take a swig of water because I'm recovering from a cough. Okay. Now the slides seem to have seized up again. Just bear with me, the screen seems to have completely seized up. David, I don't know whether you can maybe unshare my screen and then we can share it back again, because it doesn't seem to be responding at all at the moment.
Okay, this is Dave King speaking. Yes, I'm just going to, would it help, Carol, if I show my screen and then you can carry on from that?
I guess I could just let you know when ready for the next slide.
Yeah, just bear with me while I do that. Just got to find the slide you were on, 431. I think with that, just bear with me, folks. Okay. Bear with me a moment. Right. I think we're on this slide, Carol. Is it that slide? Carol, can you hear me?
Yeah, that's showing as minimized on my screen. I don't know what it's like for everyone else.
It's coming through now. I can see it enlarged on the my test screen here.
Okay. Maybe if it doesn't look right for people, they can add in the comments. So I'm ready for the ready for the next slide.
Okay, there you go.
Okay, so just to complete speaking about the reconstructed skin methods, I just wanted to make a few comments about proficiency testing. Whichever test lab you're working with, it's really important to make sure that proficiency testing has been carried out in compliance with GLP in order for any of the studies that are done for you to be fully GLP compliant. So each lab must have demonstrated proficiency in that test beforehand. And as in the OECD guidelines, they say that prior to routine use of any of the validated test methods, labs should demonstrate technical proficiency by correctly classifying the 12 proficiency substances, and they are actually listed in the guideline. So next slide, please. I'm not expecting you to be able to read the details of this. This is just to show you some sample data generated in our lab using the epidermis skin model for the skin corrosion method. And for each of the 12 proficiency chemicals, we've done the incubation at the required 3 minutes and 1 hour time point, which is shown by the grey and the pink blocks respectively. And those are carried out alongside the designated negative and positive controls, and those bars represent the percentage viability in each case compared with untreated skin model negative controls. So you can see there's a wide variety of corrosive potentials across that range of reference chemicals. Okay, next slide please, Dave. And so what we ended up with is a set of results here which proves proficiency to carry out the test. Remember, this is actually the epiderm test, so it's a bit more straightforward. It's just showing corrosive versus non-corrosive, and just demonstrating that each of those 12 chemicals of varying corrosive potential were correctly classified. So that should be available as a GLP regulatory report from the test lab. Next slide. So that really concludes what I wanted to say to give an overview of the reconstructed skin methods. If we move on now to a slightly different method, and that's OECD Test Guideline 435, and it's the in vitro membrane barrier method, which also has been around for quite a long time, for 14 years. It was first adopted in 2006 and last updated in 2015. Next slide. So the scientific rationale behind the Corrositex method is that rather than having a biological system as we have with the reconstructed skin, here we have an artificial membrane barrier which acts as a surrogate for the in vivo skin and allows you to look at damage to that barrier. And an assumption is being made that those same mechanisms of corrosion that operate on living skin would take place on an artificial barrier. And so the chemical is actually corroding the barrier, the artificial barrier. And the test system has two different components. There's the synthetic macromolecular biobarrier, and there's the chemical detection system, which is really consists of a series of pH sensitive dyes. It's quite a simple system just to look at how a chemical manages to break through the membrane barrier, and then you would measure a change in the colour of the underlying solution and match that up with a chart to show the amount of breakthrough that has been seen. Note that there's currently only one commercial test kit available for this particular method, so in a sense it's more of a simple decision compared with the skin model methods, and that method is Corrositex, but you won't see a reference to that in the guideline itself. And this method also is suitable for substances and mixtures, and it's regularly used for solids, liquids and emulsions. Next slide. So if you think about the predictive capacity of this test, this also has a fairly solid performance in relation to the Dray's test. So we've got a sensitivity of 86%, a specificity of 68%, and an overall accuracy of 81%. Now this test is, I think the big advantage of it is that it is able to identify non-corrosives and all of the subcategories of corrosive. So you will get a definitive output of subcategory 1A, 1B or 1C. However, it's got quite a limited applicability domain. There are only certain chemical classes that it's suitable for, and these include organic and inorganic acids, acid derivatives and bases. Next slide, please. So if we think about what the strengths and weaknesses are of this particular method, once again, it has formal validation and OECD approval. As we've just said, it allows full subcategorization into 1A, B, and C. And also it's a simple test method with a quick turnaround. It doesn't involve a need to wait for in vitro skin models to be manufactured and those cells to form a differentiated epidermis. It's a biochemical test essentially, and so it's much quicker to perform. But it's also a weakness that this test has no cellular components, because we're not actually testing on a living system, we're not testing on real living skin. And also in some cases, some of the colour changes that are seen in practice can be transient and quite difficult to interpret. Even though reference photos are provided by the test developer, it still is not quite as simple as it sounds and does require a trained eye to make sure that the right classification is observed. As with the other methods, it's not validated for gases or aerosols. And just to keep in mind, it's usually not applicable to chemicals with a pH between 4.5 and 8.5, because they won't be detected by the pH sensitive dyes that are used in the chemical detection system. I think in the EU validation study, it was 58% of the test chemicals were found to be incompatible with the method. So even though this looks like an attractive method for subcategorization purposes, you'll quite often find that your chemical may not be compatible with it. Next slide. So this test method, as we've said, is only valid for a limited applicability domain. As with the other methods, it doesn't discriminate between the skin irritants category 2 from no category, but you could go on to use Test Guideline 439 for skin irritation if you needed to go down that route. Next slide. So finally, I just wanted to say a few words about the third test guideline, which is 430, and that's the transcutaneous electrical resistance method, which was also adopted back in 2004. Next slide. You'll see this big pink note on the slide. I just wanted to make it clear that because Accelerate is a completely animal-free testing lab, that this isn't a method that we actually perform at Accelerate. So I wouldn't want to imply that we do that. As this is an educational webinar, clearly I wanted to make sure that we're putting all of the methods in here to discuss today. So the scientific rationale behind this method is that it's a readout of the erosion of the stratum corneum. And the theory is that when we place a corrosive chemical onto the skin, the barrier is damaged, which then causes a change to the transcutaneous electrical resistance of that barrier. So skin discs are taken from rats, and the test material is applied in a 2-compartment system for times up to 24 hours. And then the corrosive materials are identified by their ability to change to cause a reduction in the TER. And this method is again suitable for both substances and mixtures, and regularly used for both solids and liquids. Next slide. The performance of this test compared with the DREs, the sensitivity was 88.1, specificity 72.4, and the overall accuracy 79.4. In terms of the GHS categories, the test is able to discriminate skin corrosives, i.e. category 1, from non-corrosives, but it is not accepted for identifying any of the subcategories 1A, B or C. So in my view, this test is utilising animals, but it's not actually providing a scientific advantage in terms of subcategorization. Although I know there are instances where it's still used. Next slide, please. So the strengths are it's formally validated, and because it has a different mechanism from the other 2 tests we've talked about, it may be considered to provide complementary evidence. However, it's important to note that the IATA states that all 3 are considered as standalone methods, so you don't necessarily need to do more than one. The weaknesses are that the method does require animal sacrifice, and in fact in some territories the IATA states that it may be considered an in vivo animal experiment because of the preparation that is required prior to that animal sacrifice. So the stages of shaving, washing and treating the animals may be considered a live animal experiment. So that's just important to keep in mind. It's been shown that the TER cutoff value can vary with the age and strain of the rats, and as with the other methods, this isn't validated for gases or aerosols. Next slide. So just to sum up, there are some limitations of this method. It's unable to subcategorize, and just for completeness, just to state here as well that this method also is not able to distinguish between category 2 skin irritants or no category. Next slide. Next. Thank you. So I've just got a few slides just to sum up, really, having looked at the 3 methods that are in regular use for in vitro skin corrosion. And you will have seen that throughout I've stated the predictive capacity of each of the tests, and I thought it might be useful to sum those up in a single table at the end here. So you can see that the RHE method is actually the strongest performer overall, although there is a fairly solid performance from all 3 methods. These parameters were all calculated using the Dray's test as the reference. And I think it's important to note that when the animal tests were originally developed, it wasn't possible to do this kind of validation because there wasn't a previous standard or a previous benchmark to validate them against. And so what's happened is over the years, industry has gathered a huge amount of experience using these tests and a level of confidence in them. And so we're never dealing with a perfect benchmark because the benchmark itself has its limitations. But having said that, I think for the purpose of building confidence in the in vitro tests to show that they are comparable with a method that's been used for decades, it still has a value. Next slide. I just wanted to just point out that sometimes skin corrosion testing might not be essential if you are able to establish a weight of evidence to show whether something is a corrosive or a no category. But for us, in terms of our experience working with companies, for many, the CLP regulation is a key consideration for many companies that are commissioning these tests. And as many of you will be aware, there are a number of logistics and cost implications for transport, labelling, and the packing groups that are assigned according to the subcategories. And it's very desirable to be able to achieve the least harmful subcategory possible in order to minimise things like transport costs, etc., and a number of other commercial factors as well. So sometimes in vitro corrosion tests are commissioned and they are considered desirable, even if a basic classification of yes, no is already known. Next slide. So if we just finish off by thinking about when an in vivo test might still be a requirement, as I'm sure you're all aware, the key regulations, including REACH, CLP and BPR, all allow animal testing only as a last resort. And REACH states that an in vivo test should only be performed in case the in vitro studies are not applicable or the results of these studies are not adequate for classification. So there could be a chance that the regulators might come back and consider that for some reason the data is not adequate and may insist on an animal study. The IATA states, and I think this is really powerful, in the interests of both sound science and animal welfare, in vivo testing shouldn't be undertaken until all available data relevant to the potential dermal corrosivity of the test chemical have been evaluated in a weight of evidence. So all of these regulatory frameworks are recognising that sometimes in a small number of cases they will still expect an animal test, but this has to be a last resort. And I think that the in vitro methods that are now available are so powerful that those cases where an in vivo test will be needed is minimal. Just wanted to state that in 2018 there were still 123 DRAIS tests reported by the Home Office in the UK, and that's slightly higher than the 2017 figure. So there still are clearly some cases where those tests are being performed. We're not able to distinguish between the eye and the skin DRAIS in those figures because we don't have that level of detail. And I think that it's just important to really drill down into the detail of the in vitro tests to understand what they can provide, just to help minimise those times when an animal test is still required. So next slide. So with that in mind, I just wanted to summarise the recommended approach that we would have for skin corrosion testing in vitro based on what I've described today. Please do look up the IATA if you're not familiar with it already. Probably many of you are, but it's important to follow that guidance to determine initially whether an in vitro test is required in the first place. And then if from your weight of evidence you decide that further testing is required, depending on the highest probability, your most expected result, you would then decide whether to adopt the top-down or bottom-up approach, which starts from corrosion or irritation endpoints respectively. And when selecting from the 3 approved in vitro tests, which are described in the IATA and we've looked at today, I would say that a good starting point is the RHE method, because it's been shown to have overall the highest predictive capacity and a broad applicability domain. And also it does have formal approval if the EpiSkin method is used to discriminate between subcategories 1A and 1B/C. So it is important, as I discussed earlier, to consider the most appropriate choice of skin model. If you do have a clear need for subcategorization between 1B and 1C, or for substances with an extreme pH, definitely worth considering adding Test Guideline 435 for Corrositex, but keep in mind that it does have a limited applicability domain. And if you're unlucky enough to find that your chemical doesn't apply to that, is not applicable for that test, what you could do is alternatively consider using the RHE capability in those published studies that I mentioned to discriminate between 1B and 1C for weight of evidence purposes. More information is given about that in the IATA, or you could choose to default to 1B, which is looking at sort of worst case scenario. So it's exercising caution in terms of the labelling of that material. Having looked at the evidence in the IATA, of the 3 tests, the TER has the poorest predictive capacity out of the percentages that we showed. It doesn't have the subcategorization potential, and because it also requires some in vivo steps, it's not considered an ideal choice by most labs. So next slide. So I think that's it. It's a bit of a whistle-stop tour through the in vitro skin corrosion tests that are available. I hope that's been useful, and sorry about a few technical hitches. But I'd like to perhaps hand back to Jill now and to ask if there's any questions.
Okay, so thank you all. If you have, excuse me, any questions, then it would be great if you could type those into the questions box on the GoToWebinar interface. We've got a couple of questions that have come in already, so perhaps I could start with those. So the first question then, Carol. question here asking whether or not an accredited laboratory needs to be used to perform the in vitro tests.
Well, it depends what you want to use the data for. If you need the data for a regulatory submission, it's essential that the tests are performed to good laboratory practice, so you would definitely need a GLP accredited lab for that. If you're performing the test for just new product development purposes for your own information, then it isn't essential, but would still be recommended in terms of getting the best quality data.
We've got here just asking about saying it would be interesting to see whether comparisons of the methods exist not only against the DRE's test, but are there also comparisons against human epidemiological data? And if so, would OECD 431 still look the best if you were able to compare it to human data?
I think that's a really good question, and I think there are some references to a very small amount of human epidemiological data, but clearly for an endpoint like this, it is quite difficult to gather sufficient data to be statistically meaningful, and also recognizing that most of those reports are anecdotal reports from occupational incidents, etc., which in our experience trying to look for human data can make that very difficult to rely upon and to quantify. So I definitely think it would be an amazing thing if we were able to compare all of these methods to human in vivo, but we just don't have the data available at the moment.
That's great, thank you. And kind of following up almost perhaps to that question, another question here asking, are there any examples where the in vitro methods have got it wrong?
Not that I know of, but of course that doesn't mean that there aren't examples out there. I would imagine that because the methods are validated against the in vivo DRE's test, that there's a chance that that method would have got it wrong in some cases as well. So I know that there's always been some concerns about the performance of that test, and now ironically we use that as our benchmark to validate the in vitro test because it's the only benchmark we have. So to some extent we're always working with a moving target, and we just have to make the very best assessment that we can with the information that we have, and err on the side of caution where necessary. I would say though that for a basic endpoint like skin corrosion or irritation for that matter, the biological pathway is pretty straightforward. It's pretty basic damage to the skin cells, and therefore easier to predict in vitro than some of the more complex pathways like skin sensitization, for example, where we have the newer methods that have come out more recently.
Okay, so our next question. So your labs claim to be 100% animal free. What growth serum do you use for your cell cultures?
We use human serum for our cell cultures. For this particular type of test, they are commercially provided reconstructed human tissue models. So we're actually buying those validated models in, we're not culturing cells in the lab. So those models have not been exposed to fetal calf serum when they were manufactured. But to say we never use fetal calf serum in our lab to grow human cells, we grow human serum in those tests where it's appropriate to grow those monolayer cell cultures.
Okay, another question. So if the expectation is irritant versus non-hazardous, so the bottom-up, using the bottom-up approach, which is the best test method to use?
For the bottom-up approach, where you would start with skin irritation, that's a more straightforward choice because there's actually only one that's regularly used, and that's Test Guideline 439, using a reconstructed skin model, actually pretty much the same skin models that we talked about today. And the method is very similar, but it's been validated for irritation potential versus corrosive potential. It's using different time points, different reference chemicals, but that would certainly be the go-to method for skin irritation.
Okay, a question on eye corrosivity and irritation now. Does eye corrosivity and irritation testing have a similar IATA which can be followed?
It does, yes. There is an IATA on eye irritation and corrosivity, looking at the different in vitro methods and in vivo methods for that. So I think if you just search online, you should be able to find it quite easily, but if you can't, by all means, follow up with us later.
Okay. And another question, would Test Guideline 435, the membrane barrier test, be suitable for mixtures as well as substances with pH extreme, or is this too difficult to predict?
No, it is actually suitable for mixtures as well. So as long as the pHs fall within the acceptable range for the test and that the changes can be predicted, it's okay to use it for mixtures. I would just point out that all of the formal validation studies for any of these methods were originally done using single chemicals, not mixtures. And so that is a thing across the board. But the OECD expert working groups, when they're reviewing the data, they do also make an assessment on whether they feel that they can say that it's suitable for mixtures. And for all of these methods, the test guidelines say that they are.
Okay, that's great. Okay, we don't have any more questions that have been submitted. If you want to submit a question, perhaps get it in very quickly now. Otherwise, just wanted to really say thank you very much to Carol for a really informative and excellent presentation. So I've certainly learned a few new things today, and I'm sure that everybody else has as well. So thank you very much to Carol. We've really enjoyed your presentation. We will be ending the webinar shortly. And after the webinar, we will, as we said earlier, be sending you an email with a link to our questionnaire. We would be really grateful if you would fill that out and give us feedback on today's presentation, and also for any other ideas for webinars and so on that CHCS could be running for you as our members. Okay, so if we don't have any more questions, then that's the end of the webinar. Please hang up or close your browser windows, and we look forward to hearing your feedback.
Yeah, thank you, everyone.